Thanks to the organizers for this opportunity. What I'm going to present today is actually work by my lab as well as my collaborator's lab, Dr. Bhaumik Patel, who is a professor at Medical College of Virginia, as well as the chief of the Division of Hematology Oncology at the McGuire VA Medical Center in Richmond. We both work on cancer, especially one part of cancer, which is cancer relapse. The US especially has had a number of cases, a couple of million new cases every year for a long time. And there are more than 20 million cancer survivors today. The anti-cancer drug market is also increasing at a phenomenal rate, which gives us a phenomenal leeway into trying to discover new molecules that would hopefully be funded by pharmaceutical industry eventually, and hopefully that will reach the clinic. If you see on the right-hand side, across a panel of different cancers, the primary reason why people die of cancer is because of metastasis, progression, and reawakening of cancer after surgical removal or chemotherapy. This gives us the central hypothesis that the primary reason why we have cancer problem is because of cancer relapse. And to put it in just one line, there's actually no real drug available that could treat the fundamental aspect of cancer relapse. So at the same time, one can imagine that drugs that prevent a relapse would essentially be resolve practically all cancers, because death would therefore be eliminated. The cancer relapse arises from a particular type of cells that seed every tumor. Those are called cancer stem cells. Cancer stem cells tend to hang around primarily because cancer stem cells are not eliminated by chemotherapy or radiation therapy. The basic idea here is that cancer stem cells, back in the olden days, they used to be called cancer stem-like cells, but nowadays the terminology is gradually shifting towards cancer stem cells, which is what I'm using. So they are slow at renewal. Their cycling rate is very slow, and this is the reason why majority of normal chemotherapy drugs typically leave aside cancer stem cells. Radiation therapy exactly for the same purpose. So they lay dormant in the niche and they reactivate after a very long time under certain conditions, which to a certain extent have not really been fully delineated. So this is very similar to a bear who's actually hibernating, especially in winter, and no amount of powerful gun could really reach the cave in which the bear is hibernating, which is the reason the bear survives and then reawakens when the time is right. So we hypothesize that we should be able to target cancer stem cells using an absolutely fundamental approach. The fundamental approach relies on two unique aspects that have come about because of the collaborative nature of the work that is being performed in my lab as well as Dr. Patel's lab. So my lab specializes in generation of a diverse library of small molecules, especially that belong to the class of glycosaminoglycans. So glycosaminoglycans are natural products. They exist obviously in all of us. And the key aspect of glycosaminoglycans, as they are found in nature, is that these molecules are highly sulfated. And the negative charge is the reason why these molecules bind to a number of different proteins. There are various steps or various processes in which glycosaminoglycans participate. And these include growth, differentiation, morphogenesis, angiogenesis, metastases. A number of these processes are regulated by glycosaminoglycans. In fact, specific structures in glycosaminoglycans regulate these processes. So my lab essentially generates synthetic organic molecules that mimic glycosaminoglycan structure. So that is one aspect, and therefore we hypothesize that these molecules would be able to target the absolutely basic process of growth and proliferation at the very first step, which is at the stem cell level, because of the role of glycosaminoglycans in that process. And the second aspect is the assay that we used. The assay is shown right in the middle. We use a tandem dual screening assay, an orthogonal assay, which tests whether the molecules would inhibit spheroids or cancer or cellular growth in the spheroidal state versus the monolayer growth. And in the second step, we test for inhibition in the spheroidal state without any treatment of the drug. And using both of these processes, using this kind of an assay, we have been able to identify molecules that target cancer stem cells. And obviously after that, it's the development process, which is testing the efficacy of new chemical entities, both in vitro, in vivo toxicity, and things of that sort. So what I'm going to present today is essentially these three aspects and the discovery of a novel molecule, glycosaminoglycan molecule that targets cancer stem cells. So first, library. We now have developed a library of something in the region of 130 organic molecules that are sulfated. These are completely new. These are not commercially available. These are basically synthesized primarily in my lab. And we have published more than a dozen papers on the generation of these molecules over the past decade, or slightly more than a decade or so. The key aspect of these molecules are they are fully synthetic and they are homogeneous. This is completely different from glycosaminoglycans, which are heterogeneous and polydispersed as they are available in nature. The second aspect of novelty of these molecules is that these are fully water soluble. So although these are organic molecules, these are completely water soluble, primarily because these have a number of sulfate groups. So as you can see right in the middle across several different scaffolds, the number of sulfate groups can go from just one sulfate per scaffold to more than 10 or 12 sulfate groups per scaffold. And that engineers selective biological activity. Second, the assay. The assay, as I was alluding just a little bit earlier, consists of orthogonal dual or tandem screening assay, where, for example, on day one, what we do is we plate cells, cancer cell lines under the spheroidal conditions. And they typically grow as spheroids, fairly distinctive, 50 micrometers or so. And we add the molecule at various concentrations, for example, 100 micromolar, to observe whether there is inhibition, 50% inhibition of spheroids. At the same time, we test monolayers too, which is 2D culture, and assess whether these molecules inhibit monolayers. So we select only those molecules that inhibit spheroidal growth and do not inhibit monolayer growth. So 3D culture growth versus 2D culture growth, selective inhibition of 3D culture growth. And then what we do is, following this, we dissociate the spheroids and then replate under spheroidal growth conditions without any additional treatment of the molecule. The reason why we do this is because we want to emphasize, we want to test the self-renewal aspect of cancer stem cells. So if the cell has been able to remember that it was under inhibited conditions, then it would stay inhibited. And we assess whether under this condition, which is in the absence of the drug, we see inhibition, we identify those scaffolds. So when we did this, we have done this now multiple times. When we did this for the first time, we identified from 53 of our molecules, or glycosaminoglycan mimetics, or NSGMs, we identified 3 scaffolds, G2.2, 11.1, and G12.2. Now, the results are fairly interesting, simply from the point of view of structure-activity relationships. We find that G2.2 basically passes, this is right in the middle, basically passes both screens, screen 1 and screen 2, with very high potency, which means extremely good inhibition. Whereas a similar molecule, G4.1, which has just a change of one CH2 group, inhibits it, passes both screens 1 and 2, but inhibits it weakly. So therefore, and then G4.1, which is actually a trimer of G4, the basic scaffold, which is the monomer G1.4, 3 units. It passes only screen 1, but fails in screen 2. And the monomer, which is G1.4, fails both screen 1 and 2. This indicated that the structure-activity relationships here is very tight and gave us sufficient reason for the high selectivity of this molecule against one or more proteins involved in fundamental aspects of cancer stem cell renewal. So here is the conclusion. At the end of our first screen, we had in the second group, we had 11 molecules, and then we identified G2.2 as the primary molecule to follow. We eliminated other molecules because of some toxic considerations, which I'm not presenting here. Now, G2.2 is very promising. The reason why it's very promising is because it's able to inhibit colorectal cancer cell lines, several of them. It's also able to, this is in the spheroidal growth and not under monolayer growth, pancreatic cancer stem line, cell line, and under renal cancer cell line. And if you see, there are various types of mutations in these, and majority of these mutations are no barrier for G2.2 to work. G2.2 specifically impacts self-renewal, primarily because the inhibition, it continues to inhibit even at the secondary screen, which is step number 2. And then if we replate those under spheroidal conditions the third time, which is a tertiary screen, or the fourth time, or the fifth time, or the sixth time, or the seventh time, it continues to inhibit. It doesn't simply lose its inhibition, memory, indicating there is some loss of that as the passage increases. But basically, this indicates that this molecule is very likely to target self-renewal property of cancer stem cells. G2.2 can be very easily synthesized. This is a molecule that can be synthesized in 5 steps, starting from natural product quercetin, first protection, followed by conjugation to generate the dimer, then deprotection of the MOM groups, and then sulfation. The challenging step here is sulfation under microwave conditions, and this is the only step that is really challenging. Otherwise, majority of these steps are very simple. And finally, sodium exchange to yield a very fine powder, which is essentially white and very clean on ultra high performance liquid chromatography. G2.2 has very favorable CMC properties. So one, synthesis is short, purity is high, batch-to-batch consistency is also very high. It's a hygroscopic powder, so you really need to keep it safe. Solubility is pretty high. It's fairly temperature stable, so it exhibits a very good shelf life, somewhere in the region of easily a week or more. And then pH stability is also very good. It's fairly stable between the pH 5 and 10. It's also very promising from the point of view of toxicity. So if you give this molecule a repeat dose at 200 milligrams per kilogram for 3 weeks, essentially healthy animals, so no gross toxicity. There is hardly any reduction in the weight. Serum chemistry in terms of vital organs, liver and kidney, essentially show no impairment. There were no significant changes in serum electrolyte or glucose level, except for prothrombin time, which is because these molecules mimic heparin, which is an unfractionated, which is an anticoagulant that we tested for coagulation properties. But there was a mild elevation in prothrombin time, which is clinically insignificant. And actually in cancer, a little bit of anti-thrombotic activity might actually be beneficial. So here are these results. There is no overt toxicity, as you can see, through histochemical staining. In addition, The progenitors, which could also be present in the 3D culture, which is the spheroidal culture, they do not exhibit much effect at 3 to 4 times higher dose of G2.2. And likewise, we asked the question whether adult hematopoietic stem cells would exhibit. So these are organoids that are grown, and basically G2.2 exhibits essentially no effect on adult hematopoietic stem cells. This molecule is fairly efficient in xenograft model. So this is under the consideration that if G2.2 was to be used alone, then what would happen? And this would be under the consideration that you have a growing tumor. So this doesn't really test for relapse, but this is simply the growth of tumor. And we do find that G2.2, especially in several different cell lines, 2 cell lines are shown here, HD29 and HCT116, which are both colorectal cell lines. There is a significant reduction in tumor volume, tumor weight, because of G2.2. And it is also concentration dependent with regard to HCT. Note that both of these cell lines are at least HCT116 is KRAS mutant and P53 wild type. The molecular mechanism of action involves primarily a receptor tyrosine kinase. So back when we designed this molecule and we were testing for the entire idea of targeting cancer stem cell, this is actually what we were hypothesizing in terms of the molecular processes. So for example, when we have growth factor receptor on cell membranes, binding to growth factors and dimerizing to yield an activation signal, for example, fibroblast growth factor receptor, this is aided by the proteoglycans consisting of the glycosaminoglycan chain on the cell surface. And the glycosaminoglycan here brings the growth factor and growth factor receptors together in dimerization. So that is the reason why natural proteoglycans or glycosaminoglycans aid growth. But we hypothesized that glycosaminoglycan mimetics, such as small molecules, example G2.2, would bind either to the growth factor receptor or would bind to the growth factor, thereby disengaging the glycosaminoglycan chain and therefore preventing dimerization and exhibiting inhibition. So this is essentially targeting the growth factor receptor yin-yang interaction. Now G2 in terms of cell surface, in terms of cancer, cell surface, there are a number of different growth factor receptor tyrosine kinases. For example, FGFR, VEGFR, c-MET, ERBB, PDGFR, and several others. All of these receptors and their oligomerization, dimerization, for example, is aided by the proteoglycan directly, for example, as in FGFR, or indirectly, for example, in PDGFR, where the glycosaminoglycan simply serves to capture the growth factor, the ligand, which then moves over to the receptor, the PDGFR, and induces dimerization. So we asked the question whether G2.2 would actually engage any of these receptors. So what I'm not showing you here is all the intermediate studies, and only showing you that the results for phosphoreceptor tyrosine kinase microarray, which basically pinpointed that c-MET was undergoing significant activation with G2.2, which is the dimer, but with a monomer, there was essentially nothing. And IGF-1R was inducing significant inhibition, monomer and vehicle showing essentially the similar results. So either it was c-MET or it was IGF-1R or IR. And IGF-1R has not really been known to bind glycosaminoglycans. So then we performed time dependence of activation and found that c-MET essentially lost activation fairly quickly within 30 minutes, whereas IGF-1R, the inhibition of IGF-1R was sustained for a fairly long period of time, indicating that most probably G2.2 works through IGF-1R and not through c-Met. So we did mutagenesis of IGF-1R. I'm not showing you a whole lot of molecular modeling and hypothesis-driven studies. We identified 3 mutations that could actually play a major role, such as 138, arginine 138, arginine 252, and arginine 107 as the key residues. They may be involved in mutations. So we express these mutants and then measured the binding affinity. It turns out that as compared to the recombinant wild-type extra cellular domain, R107H lost some activity, and R138A and R132C completely lost activity against G2.2, which clearly indicated that these residues are engaged in IGF-1R by G2.2. So we now believe that this molecule works through IGF-1R. This is one of the first in the field, primarily because IGF-1R has not really been known to measure, has not really been known to bind to glycosaminoglycans. The IGF-1R is a constitutive dimer, and this particular molecule exhibits a major conformational change to undergo autophosphorylation and then signaling intracellularly. This particular process is prevented by G2.2, which is actually a mimetic of HSO6. The study on HSO6 IGF-1R has just recently been published in Angewandte Chemie, which you might want to download from the web. So then we started asking questions as to whether there is a relapse thing that could be targeted. We designed a very sophisticated experiment where we basically exposed animals first to Fuox, which is the chemotherapy, then waited for some time, and then we randomized for G2.2 or vehicle. And as you can see in the figure down below, that when animals are exposed only to Fuox, which is the anti-cancer agent, the relapse grows, which is a green line. It actually grows much faster than the regular control, indicating that cancer stem cells come back really fast after the clinically used chemotherapy, but G2.2 prevents it significantly in the second arm, indicating that there is a strong possibility of G2.2 likely to work in cancer relapse. And finally, we performed an experiment wherein we took cancer cells, regular cancer cells, and exposed it to Fuox under petri dish conditions, under regular growth, and we killed more than 99% of cells using Fuox. What remained was just a very few spheroids, which we then injected as xenografts into the animal. This is essentially how it works in human beings. People are treated with chemotherapy, and then after a very long period of time, the cancer reawakens. So this is essentially a simulation. Reawakening in human beings takes years, so it's very difficult to do this experiment in animals. But this is how we simulated cancer relapse, and we then split this into two parts, cohorts, vehicle and G2.2, and we measured for. And what you can see on the left-hand side is that the vehicle group, all animals died just within about 12 weeks, but with G2.2, the animals survived more than 26 weeks, which indicates they lived for several years after cancer. Human beings would live for several years after cancer. If you really check for the injection site, then in the vehicle, you can clearly see cancer growing back, but with G2.2, there was essentially no growth in cancer. So overall, what I presented is that G2.2 appears to be the first agent that selectively targets cancer stem cells, not adult stem cells, nor hematopoietic stem cells. G2.2 is a first-in-class agent, which is something that I did not present. What happens within cell, it targets P38 MAP kinase. This is a paper that has been published in 2019 in Molecular Cancer Therapeutics. G2.2 is synthesized from very inexpensive raw materials in just 5 steps. It displays no toxicity to organs, and we are currently performing advanced in vivo and preclinical development studies at this time. Finally, this is my group. I have a fairly, very active and enthusiastic group of students and postdocs and research scientists. The work that I presented today was by Dr. Nagarajan, Dr. Villuri, Dr. Afosa, Mr. O'Hara, Mr. Chidam, and Dr. Sankaranarayanan. So this work came about because of the contributions of this particular group. And finally, our lab is funded by the NIH, especially NHLBI. We have been funded by the American Heart Association, the Mizutani Foundation for Glycoscience, and 2 local chapters, which are Massey Cancer Center and Virginia Commonwealth Health Research Board. Thank you for your attention.